to you tonight. So my name is Andrea Sboner, and today we're going to talk about precision medicine and how we can translate scientific discoveries into patient care. But first, we'll start with the definition of precision medicine. There are many. The one that I like the most is finding the right drug for the right patients at the right time. And why now are we talking about precision medicine? Well, a few years ago, there has been a revolution in genomics, molecular biology. As you know, genomics is the study of genes, their function, and the techniques that we use to understand genomics. Our cells includes their nuclei, the DNA, which encodes for their entire information required to create molecules of life, or the proteins. You can think of our genome as our recipe book, where all the recipes are included, and then when you want to bake a cake, you just maybe extract one page of it. This is done through messenger RNA. It makes a copy of parts of the instruction of the genome. And then finally you get your protein or your cake. Importantly, understanding how this genome is defined helps us identify when something like a little typo in your recipe makes you create not a very good cake. So precision medicine benefits mostly for some of the major advancements that we have in what we call sequencing technologies, or NGS. And in 2003, there was the first completion, or let's say the second draft of the human genome reference sequence. You can think of it as our book of recipe for the genome. Immediately, so these human genome reference sequence is about 3.2 billion nucleotides or bases information that describes how nucleotides are distributed on our genome. It cost almost $4 billion, had an impact of almost $800 billion, and it lasted 13 years. Immediately, a lot of projects started to understand differences between populations, like HABA projects, and others, like the ENCO projects, focused on identifying the functional elements of the genome. But it was in 2007 that we had these major technological advances with the first massively parallel sequencing technologies, or next generation sequencing, or NGS for short. We sequenced, again, the human genome, the same 3.2 billion bases, but only in 4 months compared to the 13 years of the first one, at a much lower cost. Some of you here are in the engineering department or interested in more engineering. You might have heard about Moore's Law. Moore's Law states that the number of transistors on a chip might double every, let's say, 18 or 24 years, and this is what we have seen. But the point of Moore's Law is saying, usually describes something that evolves very rapidly. In the case of the genomics, we've seen the cost of the genomes going down following Moore's Law in the first 7 years of this century, and then once the new technology was introduced, the cost of genome decreased even faster than Moore's Law, which is quite incredible. That's why, starting with this new technology, we were able to expand our knowledge in both normal variation across population, for example, with 1,000 genome projects, and also apply this technology to disease, in particular cancer, which is a genomic disease. We had several projects, international projects, to study cancer and how cancer develops and progresses. But basically, sequencing technologies, what they do is they are able to provide us information about the content of our DNA or our genome. And the content is very simple. It's a series of simply 4 letters, A, C, T, G. And you can think of a sequencing technology as simply a translator between biological materials on one side and a common language of ACTG on the other side. How do we do this? There are many ways to use sequencing technology to study biology. This is the most common. So the machines will provide you with a lot of sequences, typically millions of these sequences that represents these fragments on the genome. The first step is to identify the samples that you come from. Then we align them to the chromosomes to identify where on the chromosome they are. And finally, depending on your experience, you might analyze those results in different ways. Here I'm showing one example, which is the in cancer genomics, where we've tried to identify variations or alterations in the genome that are present only in the tumor. What kind of alterations? Some are very small, like typos or little insertions and deletion of a few letters. Others are bigger, involves bigger chunks of DNA that are amplified, deleted. You can think of it like missing a paragraph or scrambling your pages of your recipe book. So by using this technology, we can identify all these alterations in cancer, for example, and that's led to the discovery of targeted treatments. These are drugs that now target specific alterations that occur in the genome. One example is in breast cancer. HER2 is a gene that is typically amplified in a subset of patients with breast cancer, and now there is a drug that can target that alteration. It's very successful. More recently, even, you have drugs that can target, that can be applied multiple cancers, as long as the patient or the cancer they have a particular alteration, in this case a rearrangement of a drug. A very promising treatment options for cancer patients is immunotherapy. In immunotherapy, we don't target directly the genomic alterations, but we harness power of the immune system of each patient to target their own cancer cells. However, genomics is still helpful to identify patients who benefit the most from immunotherapy. So over the course of the years, and by doing a lot of sequencing, we learn a lot about how cancer develops and progresses. One thing that we observed early on was this long tail of oncogenic drivers. Oncogenic drivers are genes and alterations that promote the growth of cancer. And we realized almost immediately that there are very few genes that are common across cancers, but many alterations are specific to either a cancer type or even subtypes or subset of patients within a specific cancer type. This caused a problem in terms of developing and identifying drugs that might target this long list of gene and alterations. So one way to approach this issue is to use organoids. Organoids as patient avatars, you see here. So how does it work? A cancer patient comes into the hospital, their tumors get sequenced, a sequencing report is released to the clinicians, the treating clinicians. They can start treating the patient according to the genomic landscape. At the same time, the tumor is collected, cells from the tumor are collected and growth in vitro for several options. We can do drug screening, we could, or co-clinical trials, where we treat these organoids with the same drug that the patient is receiving to see whether they would develop mechanism of resistance that could inform future treatments of the patient. We can use them and implant them in xenograft, mice, and become xenografts and do in vivo drug validation. Importantly, we could do high-throughput drug screening. We could test simultaneously multiple compounds of these organoids and find out what is the best drug or best combination of drugs that might work for a particular patient. So, over the years, we have made a lot of progress in cancer, in cancer genomics and cancer treatment, but where are we now? Here is a snapshot of the world population, and this ties to some of the talks that we heard before. Genomics databases, which are the resources that most cancer genomics uses to identify what are the alterations that are important, are heavily biased at this point on European populations. And this lack of diversity in genetic databases hampers the research and the development of new drugs and understanding of alterations that might be specific of non-Caucasian populations. At the Institute for Precision Medicine, White Cornell Medicine, fortunately, is located in New York City. It's a snapshot of diversity. Next year, we're going to expand our genomic testing at the Brooklyn Methodist Hospital. Brooklyn is quite diverse, has more than 50% of African American population there. We're also expanding into Queens, which is even more heterogeneous population. We hope that with these steps, we are able to fill some of the gaps in the knowledge about cancer in all the population. But the key point of precision medicine that I want to bring it to you is sharing information, sharing experiences as well, is the only way to make sure that we can fulfill the promise of precision medicine for everybody. The Institute is also part of several international collaborations, national and international. The All of Us project, or the Precision Medicine Initiative, whose aim is to sequence 1 million Americans. Polyethylene 1000 is another project which is New York City based, with the aim of focusing on patients and individuals from a diverse background. We are part of the International Cancer Genome Consortium with the aim of sharing cancer information, clinical information, and find new drugs. And the White Cornell is also a designated site in New York for the cancer model developments with our organoid facility. These data and genomics data are predicted to be to surpass even astronomy and YouTube videos in a few years, such that we might change our way to refer to huge numbers with the genomical instead of astronomical. All this amount of data requires different application, different tools, advanced technology to understand the content and this complex information. Artificial intelligence, we heard also earlier in the evening, is becoming more and more important in the medical field, in particular for classifying or detecting, let's say, early melanoma, or using histopathology images to identify subtypes. But also, linking back to the genomics, we could use images to understand or to predict what kind of genomic alteration they have, and therefore get better treatment faster. We are also exploring ways to represent complex data in a more understandable way. Here I'm showing a couple of examples using mixed reality. I don't know if the video works, it doesn't matter, to understand those data. So mixed reality goes from everywhere from virtual reality, that you have an example on the left, where in a fully immersive environment, you can see the impact of genomic alterations on a 3D representation of a protein structure. On your right, there is an example of augmented reality, where we overlay virtual objects in the real world. In this example, there is a cancer drug network. There are other applications that can be used in this setting. In conclusion, what I want to say is that I think to fulfill the promise of precision medicine, to find the right drug, to the right patients, and at the right time especially, we need a combination of all, mixed reality, artificial intelligence, and genomics, and organized, and also the expertise of a diverse workforce to really achieve that. And so I want to conclude just thanking all our patients that help us reaching this knowledge. This is a snapshot of the computational team at the Institute for Precision Medicine, and thank you all for listening.